Hello, everybody. Welcome to today's webinar co-hosted by uBriGene's and Circurna. My name is Jeff Heng. I'm a VP of Commercial Strategy at uBriGene's and a moderator for this webinar. I'm very excited about this webinar, not only because of this very important topic, but also because of the esteemed panel of speakers and panelists we have assembled. And we have two keynote speakers, Dr. Scott Patterson and Dr. Robert Rickert. And then Dr. Peter Weinstein will join me for a fireside chat. So without further ado, I'd like to invite Scott to give a first presentation. Dr. Patterson is trained PhD in Purdue University, and currently he's the Senior Director of Business Development, and he will present on behalf of uBriGene's on circular RNA manufacturing process development. Without further ado, Scott.
Thank you, Jeff. Yep, so just a quick reminder of our agenda. Jeff carried out the introductions. I'll talk about uBriGene's RNA manufacturing platform and how we're using it to support this partnership with Circurna, and then Robert will provide a deeper dive into the benefits of circular RNA and some of the innovations they've developed around designing and applying it for clinical application. And then, like Jeff said, he and Peter will lead the fireside chat before we field questions from the audience. So just one slide about, kind of a high-level view of uBriGene. So uBriGene is a cell and gene therapy CDMO. What we have done and what we differentiate ourselves with is we provide an integrated solution for therapeutic developers from concept to the clinic. And that includes the platforms listed on this slide. Obviously, most of the cell and gene and genetic medicine platforms start with plasmid. And so we have a great platform that I'll talk about for GMP plasmid manufacturing. And from that starting material, we're able to manufacture and QC non-viral approaches like the circular RNA program that we're partnered with Circurna on. We have the ability not only to manufacture the RNA payloads, but also the ability to manufacture the LNP formulations prior to using them clinically. And then we also are able to manufacture viral vectors, and that won't be the subject of our webinar today, but you can go to our website and view past webinars and content about that capability as well. And we use these assets, these non-viral assets, like the ones we'll talk about today, as well as the viral assets, both for ex vivo clinical applications, as well as direct in vivo applications. And another very important aspect of what we bring to our development partners is a very broad expertise and great platforms internally for quality control. So we try to minimize the amount of contract testing that has to be outsourced so that we can keep programs on budget and on time. So let's talk about our RNA manufacturing platform at a high level, and then specifically around the ability to support circular RNA programs like Circurna's. As I said, we have a very comprehensive RNA manufacturing platform, as we have for our other platforms. It starts with plasmid. So we are able to do master cell banking of the appropriate E. coli strains. We're able to do GMP manufacturing. We're able to prepare the plasmid DNA for in vitro transcription. And then we have the capabilities that we'll highlight during my portion of the presentation to be able to do in vitro transcription and circularization in a scalable way. And we have some very innovative solutions for purification. And then, as I mentioned previously, we have the in-house analytics to be able to monitor production during the process development, but also to do lot release of the final drug product. And we'll talk about some of our innovations specific to circRNA in that space. I'm not going to talk a lot about our LNP formulation capabilities, but we have a great platform for being able to take those RNA payloads. In the case of the Circurna partnership, their circular RNA and their novel lipids that Robert will talk about, and develop the LNP formulation and turn that into a GMP ready manufacturing process for making final drug product. And then at the far right-hand side here, obviously we have the ability to do aseptic fill finish and drug product release testing. So let's talk a little bit about what some of the specific challenges are for circular RNA manufacturability, because it is a tough one. And so I'm excited to share with you some of the updates that we've developed in this partnership with CircRNA in order to overcome some of these circRNA manufacturing bottlenecks. And so I'll highlight 4 of those. First of all is plasmid supply chain risk. So anybody who's been in the cell and gene therapy and genetic medicine, even biologics fields, knows that if you don't have reliable GMP plasmid supply, your program can be derailed very, very early. And so we'll talk about our very reliable and robust and the clinical track record we have with our plasmid platform. Obviously, you need great upstream processes for doing in vitro transcription, as well as in the case of circRNA, circularization. You know, if you don't start with high quality input, you're probably not going to get high quality product on the back end. And so we'll talk about that. The other really, really big manufacturing challenge for circRNA is efficient and inexpensive, scalable downstream purification processes. And so I'll talk a little bit about the unique solution that we've developed for that aspect of circRNA manufacturability. And then lastly, if you don't have great analytics in place to be able to measure the asset, you know, in a selective and quantitative way, it's hard to know how good your process is. And so we have a great assay development team, and that team has developed some innovative analytics to specifically measure circular RNA in a very reliable way to help with both in-process testing as well as final drug product release. And so the rest of my module in the webinar is going to address each of these 4 bottlenecks and the solution that uBriGene brings to solving those problems. And it starts with plasmid. So I'm going to start with the bottom box. This is our most mature GMP platform. We now make in excess of 100 GMP plasmid batches a year. We do this across multiple manufacturing sites, which is another supply chain de-risker. You know, if you have a problem at a specific facility, we have other facilities that can pick up the slack. Just track record, you know, with the regulatory agencies, we have multiple drug master files in place around plasmids that have been designed and manufactured by the uBriGene team. And we've supported in excess of 20 global INDs that have utilized GMP plasmids coming out of the uBriGene plasmid platform. And the reason groups like working with us, and the reason we've got this much business in the plasmid, is the innovations that are listed up top. So we have a terrific upstream team that uses single-use fermenters, has developed optimal fermentation processes and E. coli strains that routinely produce more than 1 gram of plasmid per liter of culture. These are all animal component and antibiotic-free processes, and they're very scalable up to 200 liter if necessary to support, you know, late-stage clinical commercial manufacturing. And then we have an equally impressive downstream team that's created and implements some great innovations in the downstream processing. We have a 2-step chromatography process that's very, very robust and very effective at both removing impurities and generating a high percentage of supercoiled plasmid with terrific recovery rates. So if you'd like to learn more about our plasmid platform, feel free to ask in the Q&A, and feel free to visit our website to learn more about that. But it's really is an important component of all of the genetic medicine and CGT programs that we support. So the second bottleneck that I mentioned is upstream process. So with regard to circular RNA specifically, you know, we've established and implemented design of experiment, and the uBriGene team has developed a proprietary, you know, licensing-free circular RNA process. This is just a taste of, you know, 10 conditions that we evaluated, and you can see that the results, the outcomes are kind of all over the board, from processes that came out of our DOE that did not give great product to processes that are routinely giving 50% plus. And in the field of circular RNA, if we can routinely get 50% of the input RNA turned into circular RNA, that's good. And so that's been kind of our benchmark, is develop a process, and we now have that. And it's been valuable to develop that in support of the Circurna partnership. And we routinely now are able to get 2 mg per mil of IVT RNA. We utilize high fidelity RNA polymerases to ensure minimal contaminants such as double-stranded RNA, which is a major impurity challenge for any RNA program. As I mentioned, we routinely get greater than 50% circularization efficiencies. And very importantly, if we're going to support clinical programs, is it has to be a scalable process. And our process for in vitro transcription and RNA circularization has been shown to be scalable from tens of micrograms of input RNA to being able to generate grams of circular RNA. And this is just a sample plot, and I'll talk more about our analytics development that has gone into supporting this program and other RNA programs. But we have a terrific reverse phase HPLC analytical assay that enables us to very selectively and quantitatively measure circular RNA coming out of our upstream process. And we'll talk more about that in a few minutes. The third big bottleneck, and this is, as is shown in the all caps there at the bottom of the bubble, it is the single biggest hurdle to circRNA clinical impact and circRNA commercialization. The downstream purification can be very, very challenging with this modality. There are a lot of processes that leverage GMP enzymes. So enzymes are a major challenge in terms of technically developing the process, but also they're exorbitantly expensive. So if you have a lot of GMP enzymes that are needed in order to execute purification, it makes for a very, very expensive process. And so what you're seeing on the far right-hand side is an example of our scalable process. This particular run was generating in the 20 milligram plus of circular RNA, and we're able to do this independent of enzyme. So this was a really, really key innovation that we made and that Circurna demanded of us. And it wasn't easy, but we have an enzyme independent process that's very scalable, very robust. And because we're not dependent upon GMP enzymes, it's extremely cost effective. And so, we're happy to talk to you more about what that looks like under NDA if you'd like to learn more about that innovation that we've brought to bear in order to successfully execute this partnership with Circurna. And then the last bottleneck that I'll address is analytics that I mentioned in that previous slide. Analytics for circular RNA is a bear, just to say very honestly. There's a lot of enzymes that can be included in the process, depending upon the process, from RNA polymerase, which obviously you have to have in order to do in vitro transcription, to various DNases for cleaning up at different steps. Some of the downstream circularization processes involve ligases, some of them involve ribozymes, and there's other processes that involve even additional enzymes. So the inclusion of enzymes makes it tough in terms of analytics. There's a lot of product that comes off of a circular RNA process. So like I said, our milestone, our benchmark was to get 50% or better circular RNA. So what that means is, 40 to 50% of what you have in that circularization cocktail is stuff you don't want, from linear RNA, that was generated, potentially linear DNA template that's still around, nicked linear and fragmented RNA. So there's a lot of stuff in a circular RNA reaction product. And we have to be able to selectively measure the circRNA and not have it be complicated by the other things that are in the reaction product. There's unreacted nucleotides as well, and there's going to be chemical byproducts that also can complicate the analysis. And so what's shown on the right-hand side are some of the most common analytic approaches that have been implemented to date. The top one is size exclusion chromatography. And it's good for separating circRNA from the small contaminants, things like unreacted nucleotides, nucleotide triphosphates, and chemical byproducts and small pieces of RNA. But you cannot, with size exclusion chromatography, distinguish circular RNA from linear RNA and other products that are similar in size to circular RNA. The other common approach for doing analytics on circular RNA is capillary electrophoresis, but there's limitations that are associated with CE as well. Like I said, it's easy to distinguish the circular RNA from small contaminants and from degradation products, and even the linear precursor RNA that came out of the IVT, but it's really tough to distinguish circular RNA from nicked linear, which you're always going to have some nicked linear based on current circular RNA processes. And our team, our analytical development team, have developed a reverse phase HPLC assay that I'll talk about in the next slide. And if you'd like to learn more about that it was reported at the World Vaccine Congress about a year ago in 2025, and we can absolutely provide you with access to that poster if you'd like to learn more about it. But this summarizes the results that we routinely are getting in this Circurna partnership. And this approach using a reverse phase HPLC QC assay, which, like I said, is described in more detail in that World Vaccine Congress poster, is able to quantitatively resolve circRNA from the smaller impurities and degradation products, which are those small bumps shown at the left-hand side of the chromatogram. And it's also able to distinguish circular RNA very nicely from the linear and nicked linear RNA, which is what's shown in the peak at the far right-hand side of the chromatogram. So this is the industry-leading, cleanest, and most quantitative platform for being able to get reliable analytics of the circular RNA. And it's been really, really critical for us and our process development team in order to rapidly assess, those different IVT conditions, those different circularization conditions, and being able to quantitatively say, yeah, we're getting 20% circular RNA, or we're getting 50% circular RNA, or we're getting hardly any circular RNA. So it's been a really important asset for us in this partnership and for future circular RNA programs that we're starting to work on. And so I'll summarize my piece of the webinar with this slide that just highlights the solutions that uBriGene brings to these four, critical circular RNA manufacturability bottlenecks, and that is plasmid supply chain. So like I said, we have a terrific team and platforms in place. We routinely generate more than 100 GMP plasmid batches annually. And so basically that risk is not a risk when you work with us to execute your programs. We have done a lot of work, like I mentioned, in optimizing the upstream process, both in vitro transcription efficiency as well as the circularization efficiencies. And we have a very robust and scalable process now in place for routinely getting greater than 50% circular RNA upstream. And these last two are ones that we are really proud of, the proprietary processes that we've developed around enzyme-free downstream process purification. And the fact that we're not dependent on any GMP enzymes means it's a very reliable, scalable, and cost-effective process, and is going to get the Circurna program to the clinic on time and under budget. And then lastly, it's really important that we be able to have reliable quantitative RNA species, circular RNA species specific QC assays. And we have that qualified HPLC assay that I mentioned in the previous slide that is utilized throughout PD, throughout manufacturing, and throughout final drug product lot release. And so that's the end of my discussion about manufacturability. I'm going to hand it off to Robert now to talk more specifically about the Circurna program and what they're doing with the circular RNA LNPs that we generate for them.
Thank you, Scott, for a very informative talk on the circular RNA manufacturing bottlenecks and solutions. I like to add a housekeeping note. On the webinar platform, you are not able to speak, but you are welcome to put in your questions to the question box, and we will answer them at the end of this webinar. So now, with that squared away, it's my pleasure to welcome Dr. Robert Rickert to the podium. Dr. Rickert is a visionary immunologist and oncology R&D leader. He currently serves as the CSO at the circRNA, and previously he was a former CSO at Pfizer. And so without further ado, Dr. Rickert.
Okay, thanks, Jeff, and thanks, Scott, as well. Let's get this in presentation mode. Can you see that, Jeff?
Yes, I can.
Okay. All right, very good. So it's my pleasure to introduce circRNA, which is a Houston-based biotech company formed in 2021. And really what we're trying to achieve at circRNA is to develop a industry-leading circular RNA platform, end-to-end platform. Excited about this opportunity because we think that the circular RNA platform actually represents a new area of therapeutic development that will rival, if not surpass, other modalities such as small molecules and biologics. And in doing so, will allow us to address multiple diseases across therapeutic areas such as oncology, fibrosis, and chronic conditions such as inflammation and autoimmunity. So this is an overall view of the platform itself. So of course it starts with the circular RNAs, the single-stranded RNAs themselves. And we've developed a proprietary platform here that we view as a universal vector that will allow us to introduce single or multiple payloads. As you just heard from Scott, the next step, which is really quite a considerable hurdle that we've overcome in collaboration with the uBriGene group, is to purify, well, first scale and then purify these circles at high purity such that we can effectively encapsulate those into lipid nanoparticles and continue to move these into clinical settings. So the lipid nanoparticle itself has a few characteristics that we view as important and also distinguishing to our platform. One is thermal stability. So to eliminate cold chain storage, our lipid nanoparticles can be stored extensively at 4 degrees and even at room temperature. And then to direct these LNPs appropriately, so to the appropriate tissue or even to the appropriate cell type. And then lastly, we're in a unique position via our collaboration licensing with Kindeva to not only deliver these LNPs via intravenous injection, which is common in the field, but to have the unique capacity to deliver it to the dermis via microneedle patch. So I think you can immediately see the benefit of that in the context of vaccines, where you deliver it directly to the immune system, that is the dermis, where both lymphocytes and myeloid cells reside, and then can be delivered to the draining lymph nodes and systemically. But we think that'll also be critical for oncology applications, dermatology, autoimmune applications, and even for transducing T cells in the context of CAR T. So we're excited about circular RNA. This is just to compare to the existing linear RNA platforms, which you may recall was a platform used in the COVID vaccines and was effective, but certainly room for improvement on multiple fronts, especially as we gear up to apply it beyond vaccine development. So of course, circular RNA itself, shown here in the left column, is unique in its structure, its circular structure, as well as how proteins are expressed via internal ribosome entry sequences, otherwise known as irises, and then the circularization sequences or the ribozyme sequences that actually allow the linear to become a circular template. And that's compared to linear RNAs, which have a 5' cap, 3' poly A, which allows protein expression, but it also allows for recognition and degradation by cellular endonucleases, which rapidly eliminate those linear mRNAs. So the results of that process, if you compare circular versus linear, is that the circular RNAs give rise to sustained high-level protein expression, whereas the linear RNAs result in a burst of protein expression, but then as the RNA goes away, the protein goes away. And also, it's important to remember that these linear RNAs, in terms of the host immune system, are recognized as foreign entities, much like RNA virus viral genomes. So there's host cells, host immune sensors, such as RIG-I and TLR7, that can recognize these and promote an innate immune situation, which is okay for vaccines, but is not acceptable for virtually any other therapeutic application. Scott went into the manufacturing and complexity, which is, as I mentioned, a big hurdle that we're happy to say that has been overcome via our collaboration with uBriGene. So we're moving ahead into multiple areas or applications. And if you think about it, the sustained production of the protein really allows us to consider applications that go well beyond infectious diseases, including immune cell and other cellular reprogramming efforts, even gene editing and protein replacement. So to get into some data, initially much of our proof of concept was leveraged using the yellow fever vaccine situation where there is an existing but imperfect vaccine. In this case, the vaccine components are the something called the pre-membrane protein and also the envelope protein. We call those PRME. You can see in the upper row here that those are efficiently expressed in cell lines. In the linear case in blue, an initial expression roughly equivalent to the circular RNA that then wanes over time. This is showing 1 to 9 days, whereas the circular derived protein is sustained for many days, actually beyond a week in this in vitro setting. That's not unique to cell lines, but we've shown that in primary cells as well, including monocyte-derived dendritic cells shown in the upper right, where we can continue to detect expression approaching 2 weeks. Going in vivo and looking at antibody titers. On the far left is, again, the yellow fever vaccine in a hamster model where we see post-prime as well as post-boost, high neutralizing titers that rival or equivalent to the yellow fever vaccine. That's a notable achievement because the current yellow fever vaccine, while effective, it's a live attenuated vaccine, has issues with safety, in particular for older individuals. So 60-year-old are not eligible to take the vaccine. So we think that this vaccine, and excited about developing this into a vaccine that can replace the current vaccine and have a much cleaner safety profile. So moving on to hantavirus is the lower right couple of figures looking at the glycoprotein itself derived from hantavirus and showing that one gets robust titers against that target 2 weeks post-boost, that's shown in red. You do see some from a linear derived modified RNA template, but those levels of antibody titers are much lower. And when you look specifically at neutralizing antibodies, abundant neutralizing titers with a circular RNA-derived template, whereas very little neutralizing titers with the linear template. So we moved that into animal models, a hamster model where one actually challenges the animals with the live virus. And we're excited to see that the virus conferred 100% protection. So similar to the current vaccine for yellow fever. Of note, in the middle panel here, that wasn't associated with any adverse effects in terms of the health of the animals, so no loss of weight or other parameters not shown here, and virtually no detectable virus titer compared to vehicle alone. So very effective in the animal models. We wanted to see exactly how stable those RNAs were in vivo following intramuscular injection. And that's shown here for our circular RNA as well as a modified linear mRNA template. So this is using yellow fever, as I just described to you in the previous slide. So looking for PRME expression. This is linear modified mRNA versus an unoptimized circRNA template versus our proprietary C5 optimized circular RNA template. And this is at day 1. So in the lower row, you can see, hopefully, some red staining of the cells in the linear RNA unoptimized and a little bit more in the optimized circular RNA case, but it's much more evident at day 4, which I'm now showing you, where you can see that the linear RNA mRNA is signal protein presence begins to wane a little bit more in the unoptimized circular RNA, but quite robust PRME detection at day 4 with our proprietary optimized circular RNA. We're also curious, this is the muscle, so common for vaccine injections, which cells are expressing the RNAs and where they reside. So this is using the 10x Genomics platform and the Xenium microscopy approach. We see at day 1, both linear and circular RNA are equally detectable. And this is on a per cell basis, so not looking at particular sections subjectively, but quantitatively. So similar at day 1, but quite dramatic difference at day 4, where you see that the circular RNA template is stable at day 4 and even beyond. And the expression occurs, you can see the cyan staining in the upper row. Turns out that's not the muscle cells themselves or the muscle fibers, but collections of myeloid cells. And we've confirmed that. I don't show that here, but we've confirmed that to be resident macrophages, which interestingly reside at these sites for long periods of time, hence their name. They don't recirculate through the body. We think that's the primary source of the antigen over days and weeks. So looking at our current pipeline, we are excited about continuing the development of our yellow fever vaccine. This has now been spun out into our sister company called Vaxurna. Happy to see the continued advancement of our GMP material, which should be achieved later this summer, and then moving into the clinic with that program in early next year. So Q1 2027. So in addition to that independent effort, if you will, it also serves as an important proof of concept for the platform and leveraging it for other applications. And I just wanted to highlight 2 of those. The top one, CD19, CD20 for both oncology and autoimmune applications. This may be familiar to many of you, as there's a lot of agents, in particular autologous CAR T, that have shown to be effective in directing CAR T cells to B cells and eradicating those B cells. Initially in the context of oncology for both ALL and lymphoma applications. But more recently, in the last couple of years, a lot of exciting data, clinical data coming forth that one can also utilize this modality for eliminating B cells in the context of particular autoimmune diseases. It's initially lupus, but impressively, that's now been extended to other autoimmune diseases, and the durability is continuing to become established and looking very promising. We're excited about our platform for both of those applications because one can deliver the in vivo CAR T to eradicate those refractory tumor cells in the case of lymphoma that reside in particular locations or happen to have lost the target antigen, CD19, so these can be eliminated via CD20. And for the autoimmune application, if you think about these patients that are dealing with chronic disease, utilizing an autologous CAR T platform poses a risk, although its impressive effectiveness poses a risk to the patient, and that the lymphodepletion regimens can make the patient more susceptible, of course, to opportunistic infectious diseases or infectious infections. So very promising in that space. We're excited about the opportunity to deploy our approach in both of those indications. And then moving down to fibrosis. In this case, fibrosis is a multi-organ disease, or diseases, I should say. And what we're getting at here with the delivery of a particular program to a particular organ is get at the basis of the disease. So not just treating the symptoms, but really trying to ameliorate disease, lessen the fibrotic condition to really bring value to those patients and better their health condition. So that's moving along rapidly as well as we approach candidate selection. So in way of summary, we're quite excited about the platform and its potential. I showed you that it can be leveraged in multiple areas, therapeutic areas, multiple diseases. So we really view it as a therapeutic agnostic platform. We continue to optimize it in terms of proprietary sequences, be it the IRES's circularization sequences, other aspects to drive those LNPs for particular suited purposes, programs, applications, and the LNPs as well to deliver those to the appropriate tissues and cell types. And that's via a collaboration with GATC. One of the most impressive things actually about this platform, if you compare it to other modalities, and perhaps most notably CAR T as well as biologics, is the rapid development of the platform. So we can go from an idea of the construct that we want to make to synthesizing that construct, expressing it, validating it in vivo, producing the circles, and entering it into the clinic in less than 3 years, which is a pretty rapid development. And then, interestingly, we're happy to see that the FDA is quite receptive to these new platforms that will really bring a lot of potential to patients, so that we're looking forward to those discussions with FDA as we bring this forward into the clinic. And then lastly, I should add that I mentioned our internal programs, but we're also exploring opportunities with external companies in leveraging our platform for particular targets of interest on the part of external parties. So with that, I'll hand it back to Jeff to initiate the fireside chat.
Thank you, Dr. Rickard. That's such an exciting talk. And so now it's actually truly my privilege to introduce my fireside chat partner, the panelist, Dr. Peter Weinstein. Dr. Weinstein is an accomplished biotech executive with 2 doctorate degrees. One is a PhD, one is JD. And not only he guides circRNA strategy, but I would like to say that he really assembled a world-class team, including Dr. Rickard, we just heard from. So Peter, I'm so excited about this chat because I feel like we are trailblazers going through some really exciting territories to develop this new therapeutic modality. So circRNA becomes a really important area of therapeutic development, signified by a few major billion-dollar M&A events. Eli Lilly in early 26 announced a $2.4 billion acquisition of Warna Therapeutics. October 2025 sees the BMS acquire Orbital for $1.5 billion in cash. Peter, can you shed some light on the advantage of circular RNA to make it the focal point of major pharmaceutical companies?
Yeah, so I think we have to start going back to linear RNA, where this kind of started, and people's understanding and recognition of it when it really broke out during COVID. And, my recollection, my background, my doctorate from University of Pennsylvania was in immunology, studying B-cell development. So it was pretty obvious to me at the outset that the issue with linear RNA is it didn't last long enough So it didn't produce enough antigen or protein to drive an immune response. So it felt like you had to go back and get a shot every 30 minutes. Circular RNA overcomes that. So Moderna has done a melanoma study. They got what they consider positive results. The downside of it is it's 10 administrations, which realistically is not commercially practical. Circular RNA overcomes that. In our studies with 10x genomic technology, we've shown that linear RNA, most of it's gone within a day or so, that by 4 days, over 99% is degraded, but there's still a lot of circular left that stays out to 7, maybe as long as 14 days or more. So you get significantly longer durability. Our circular RNA has been engineered to produce significant protein from the circle, but we're never satisfied. We're leaders, not followers. So we have a partnership with an AI company called GATC Health to develop second generation iris protein production sequences, which we're doing now the goal with to produce considerably more protein, and we're even producing now, which affects the dose. So with circular RNA, you're looking at maybe 1 or 2 administrations. A company in China called Ribox is doing a clinical trial for patients who had head and neck cancer and radiation treatment to stimulate saliva production, and they found significant saliva production after 1 administration. Pretty much every other circular RNA company in the world, whether the United States or China, their lead therapeutic is CD19 in vivo CAR autoimmune, and all of them are doing it within 1 or 2 administrations. For linear, it's considerably more. So with regard to pharma companies, the durability of circular RNA, and in our case, in partnership with uBriGene, the reproducible manufacturability of it at a very high purity, at a very reasonable low cost is a big deal. And as Dr. Rickert brought out, it's also related to the fact that we consider circular RNA to be disease agnostic. So, kind of the motto of the company is, you show me the protein or peptide, you tell me the disease, and we'll figure out how to treat it with circular RNA and using the body to produce the protein versus biologics manufacturing and peptide manufacturing. So, you can, as you saw from our clinical profile, what we're going into the clinic with, you can do oncology, autoimmune, fibrosis, CNS, cardiovascular, pretty much any disease can be treated with circular RNA. And I believe it was the CEO of BioNTech who was stating that he believes, and I don't think it's unreasonable, I think it's reasonable, that within, let's say, 10 years, maybe as much as 30% or more of biologic manufacturing of therapeutic biologic treatments will be through RNA. And we believe circular RNA because we believe it's a very robust platform. It's not reactogenic. It's not immunogenic. And the cost is, our manufacturing cost allows us to be a very affordable therapeutic modality.
Thank you, Peter. So, yeah, both you and Dr. Rickert allude to this wide spectrum of indications in addition to CD19, CD20, and Dr. Rickert also introduced fibrosis and other infectious diseases. So as a CEO, can you share with us the company product portfolio when you decide on such a wide spectrum of indications?
So at this stage of the company development, obviously the first consideration is their market, right? You talked about JD. Before CircRNA, I had a boutique law firm working with hundreds of small companies and startups. And, I always told them, what's the first question you ask when you're starting a company? It's who's the buyer, right? Who's the payer? And so for us, we're always thinking down the line, when we develop this therapeutic, is there a market to sell it into? So that's the first consideration, which should be the first for everybody. The second consideration for us, obviously, is the disease. Which diseases do potential corporate partners, big pharma partners want to see, but also what diseases, is the greatest opportunity. So obviously oncology, fibrosis, we believe, autoimmune and such. And then the third parameter, which I think is an important one when you look at our portfolio, is every one of our therapeutic programs is basically a proof of concept for the use of circular RNA as a treatment modality. So for instance, everybody's excited about CAR T. Well, we're doing CAR T, but we're doing CD19/20, so we're going to catch whatever slips through the CD19. When you look at our solid tumor, we have a partnership now with a company where we're testing a protein that would be expressed from a circular RNA that actually works towards T cell amelioration, right? Nobody's talking about that with CAR T, but it's very well known that CAR T cells can be shut down because of T cell exhaustion. We want to ameliorate the T cell exhaustion. So, we're looking at how do we take the CAR T and even make it better than it is now, and improving CAR T, not just autoimmune, but oncology, circulating cancer cells, and solid tumors. Fibrosis, I find a very exciting study because there we're actually targeting a tissue within the fibrotic organ targeting a specific cell within the fibrotic tissue to produce the protein in situ within the tissue to treat the disease. Once we're able to show that, then there's dozens of diseases. Now you start realizing we can start producing the therapeutic protein or peptide in the tissue to treat the disease. And then obviously the vaccine. Dr. Rickard talked about the yellow fever. We spun out the vaccine infectious disease side, as everybody's well aware. Not a great time to do RNA infectious disease vaccines in the United States, into a new company called Circurna, which has a partnership with Duke New University Singapore, where they asked us to do a circular RNA dengue fever vaccine to compare to our linear. And, the control in 2 administrations, lethal dose challenge, 60% of the controls died, 40% of linear, 100% survivability of circular. So again, as a vaccine modality, we believe circular RNA is a very robust platform. And if we had it during the COVID pandemic, probably would have been a very different situation than it worked out with the linear RNA vaccines. So, I think the first thing is there a market for it? The second thing is it a significant area? And the third thing is, does it prove the multitude of therapeutic uses of circular RNA to show what it can do? And so those are the parameters we measure our decisions on what we're moving forward with in our clinical programs over the next 3 years. And the last thing I'll say too is, there's about 10 or 11 real circular RNA companies in the world. Most of them are just doing CD19 in vivo CAR for autoimmune. Some of them have a second therapeutic. We're the only one with kind of a cross-disease, robust therapeutic platform that we're moving very quickly in the clinic over the next 3 years.
Exciting. Yeah, thank you for the insightful answers to this strategic question. Now, go back to Dr. Patterson's discussion, because I'm the executive sponsor of Circurna's project. Certainly, manufacturing is dear and near to my heart. I know that the circular RNA circularization step and the purification step are 2 biggest bottlenecks. But now, when you look at this circular RNA manufacturing from a therapeutic developer, why is manufacturing such a big deal? Can you just shed some light on this?
Yeah, so before I get into that, I just want to answer why uBriGene for Circurna. It was not the only option we had. We had other companies. Personally, I knew the founders and other members from a prior CDMO that they worked at with one of the clients in my law firm that spoke very highly of uBriGene. We were very impressed with the technical team at uBriGene, and I'm not sure if I'm supposed to say this, but they had a lot of faith in us too. So they've taken some of the payment and shares in the company, which we believe is a significant bet on Circurna by them, but we've also eventually be a significant financial return for them. But I think Scott did an excellent job of identifying why circular is hard to manufacture. When you do linear RNA, you just create the linear RNA, put the end caps on, you're done. With circular, you start with the plasmid DNA, you produce the linear. We have sequences we've engineered at both ends of the circle to autocatalytically circularize, but it's not 100%, right? 50 to 60, maybe we get over 60% circularization at the outset, so there's contaminants. And it's removing the contaminants, which sounds like, it shouldn't be that difficult. But the reality is the linear RNA and the circular RNA, as Scott said, are not very different in size. You see 2 bands, but they're not very far apart from each other. The other thing is it's not just the linear, it's nicked RNA and other stuff in it. The threshold for purity is generally 85 to 90%. You need to have 85 to 90% circular. With the team at uBriGene, we've been over 95%. which we believe is potentially industry leading. Wouldn't be surprised. So it's getting rid of that. Some companies do the circularization with enzymes, as Dr. Patterson Scott explained. Enzymes are expensive. When you want to go to commercial scale, where you're talking about creating tens and hundreds of millions of doses, it's going to have a huge impact on the price. The second one is that some companies are using enzymes for purification, particularly an enzyme called RNA-SAR. We're not aware of really commercial scale GMP manufacturing of RNA-SAR at a reasonable price. It is very expensive, as Scott mentioned. And so one of the mantras we had at circRNA from the very beginning was that we would not use enzymes. And so as Dr. Patterson and Scott showed, we've developed a method to get high purity, high yield, and we believe low cost through essentially chromatography, not using enzymes. But even saying that, it's getting rid of the linear and other materials, which is very difficult. Even with the reason RNA-SAR is an issue is not just it's expensive and drives your cost per dose up considerably. It's also because it does do a lot of off-target killing circular. And I can't say what the companies use in RNA-SAR, what their yield is, but in our hands, the yield was generally under 20-25% with RNA-SAR at lab scale. So we knew that that was not optimal. But again, it's removal of the different components that are considered contaminants and doing it to a minimum 85-90% purity. And in all honesty, with circular RNA, probably the hardest part of it. The RNA itself, you can engineer sequences. The issue now is getting around everybody's patent portfolios, including Circurna's. The lipid nanoparticles, there's probably 50 companies around the world doing lipid nanoparticles now, including tissue targeting. But the manufacturing process is where companies get stuck. And, we know that Orbital's used, which was bought for 1FBM by BMS, is using enzymes. We also know that Orna, which was supposed to be in clinical trials for their CD19 vivo CAR autoimmune treatment, I believe as early as next month or June, is allegedly delayed theirs because of manufacturing issues. So, it is the greatest hurdle, and we think we've overcome it with a non-enzyme method to get rid of all those contaminants. And again, the issue with the contaminants is when you're looking on a gel, they don't look a lot different size-wise. So it's not necessarily very easy to get rid of them.
Wow, it's great to know. Just for the audience, the kind of words that Dr. Weinstein said about us was not scripted. So I truly appreciated your kindness. So now, just in the interest of time, I do want to leave some time for the Q&A. So now I would like to invite back Scott Patterson and Dr. Robert Rickert. We're going to answer Q&A now. Let's see. I'm going to just, because I open...
You're on mute, Robert.
So I'm going to read the questions from the bottom up. Do you have any data on circRNA in more proliferative cells, such as T cells or NK cells or others? Tissue-resident macrophages are known to not proliferate too much. The dilution of circRNA might bring you back to the durability problem? Question mark. I think it's more Robert and Peter's question.
I could take that. It looks like my video is frozen, but audio is working. Good.
Okay.
Yeah, so it is something that one recognizes, certainly, because the circular RNA will be partitioned upon cell division, but there are ways around it. One is to have very efficient transduction up front in your target cell type. The second aspect is to have very potent cargo, so that even as the expression wanes a little bit, you don't lose a lot of efficacy. The cell types that you mentioned, certainly resident macrophages, as I commented on, are non-proliferating for the most extent, so they can sustain production. One can target subsets. I won't get into a lot of detail here, but for T cells, you could potentially target subsets that are more differentiated, less proliferative. And then I think the other thing to keep in mind is that this isn't a one and done procedure. It is a platform where one can redose. So you could get the initial expansion efficacy and then redose with some of those target cells that have expanded a bit, or their clonal progeny, so to speak. So I think it's something to consider, but also something that can be addressed.
Yeah, and I'll just jump in a couple of points. First is that it really depends on the disease you're treating. So for, let's say, autoimmune B cell or B cell lymphoma, generally the resolution, the killing of the B cells occurs very quickly, up to essentially up to 100%, which is what companies like Orna, Orbital, Ribox, and everybody have shown. It happens very quick. So the dilution out is not going to be an issue. And then the second part, going to what Robert said about you can give additional administrations, unlike linear, and I know Robert brought this out in his earlier talk. In no animal species, including human clinical trials by Ribox, has anybody seen any immunogenicity or reactogenicity with circRNA. So, 2, 3, 4 administrations should not be an issue if that's what you have to do over time.
Thank you, Robert and Peter. Now, second question, can you please elaborate again on how and why you are independent of GMP enzyme for manufacturing of the GMP grade circular RNA? Yeah, so we probably cannot disclose all the manufacturing processes, as some of are yet to be disclosed. But it suffice to say that it's important to be enzyme-free, because due to A, the cost issue, and the B, availability of the enzyme. With that, I'm going to see if Scott, Peter, and Robert, do you have anything else to add to?
I'm sorry. If Robert, if you don't mind, I'll jump in really quickly. I think the first part of that is that essentially you need enzymes potentially for two parts. One would be the circularization of the linear RNA that's synthesized off the plasmid DNA. The other is to clean up all the non-circular RNA material. So in the first instance, as I stated before, we have a ribozyme, a circularization sequence that autocatalytically without enzymes circularizes. So we don't need enzymes on the front end. On the back end, we recognized early on, partly, because it was, a mantra, we were always thinking about cost. We're always thinking about the end consumer and things of that sort. But we also talked to RNA manufacturers. So we appreciated that commercial scale GMP was not really practical at this time. And so developing a chromatography method was preferable over that, which is what uBriGene has done. So essentially, it is a non-enzyme method, and it gives us high yield, high purity, and we still have to determine the final cost, but we believe low cost. Robert, did you want to say anything else?
No, I think you covered it. Yeah.
Okay. Thank you, Peter, for your addition. On to next question. Just came in. Is circular RNA stable in blood?
I think this is encapsulated in the LNPs. So there are some companies that are approaching, naked delivery of circular RNAs. That's not our platform, but certainly, the LNP delivery that we're developing is stable in the blood. There's tangential aspects to this question, maybe, of where those LNPs are delivered, but yeah, it's long-term stable in the blood and the circulation.
Thank you. It's enveloped, encapsulated the RNA, stable.
Yeah.
Okay. Let me see. All right. Next question. What level of final circular RNA purity can you achieve? Yeah, I think both Peter, Scott mentioned that it's a 95% purity after the GMP downstream, or so far it's PD downstream. Yeah. And it's a big deal, as Peter already alluded to.
And just to be clear, it exceeds the minimum threshold of what you need for purity, which is really 85, 90%. So it's greater than, what the FDA would require of us.
Correct. Okay. So next question is, can you just shed some light on the bottlenecks in scaling circular RNA manufacturing under GMP conditions? Yeah, so I think that's what Scott mentioned. That's really the four bottlenecks: plasmid, circular RNA circularization step, purification step, and eventually the analytics. Yeah, so I think the Scott has answered really well in the presentation. Okay, last one. What are the key considerations when transitioning from research scale to GMP manufacturing for circular RNA? I think that's probably more Robert and Peter's question.
I'll let Robert go first if he wants.
Well, actually, I think it was covered by Scott a bit as well. So you can add to it.
Yeah. So I mean, I think, to be honest with you, it's why we're here today, right? We're a small, company. We're out doing a Series A raise now for 100 million, which we think will close in the next 2.5, 3 months. But even then, we're not a CDMO manufacturing company. So I think the first step for a company like ours was finding a CDMO like uBriGene who had the ability to do circular RNA, which I can also tell you was not easy to find. A year ago, nobody even knew really what it was. And maybe there were 3 companies in the world willing to talk to us about doing this type of manufacturing. uBriGene brought a lot of experience into it. But I think it's really a company that has a track record and experience manufacturing GMP and GMP scale, GMP material at scale that I think is the key. And that's, for us, that's what uBriGene brings. And so I think when you're looking at manufacturing a commercial scale, let's say 10 million doses, you have to find someone who's able to do that and credibly do it, which right now for circular RNA, you probably can count those companies on one hand.
All right, to the audience, again, this is not scripted. So I owe you a dinner, yes. Peter.
Yeah, no, I mean, just to be clear, it isn't scripted, and people who know me, I'm a straight shooter. I just, say what I think is real, and we didn't come to uBriGene, say, okay, now we have to go find a manufacturer. Let's go to uBriGene. We did talk to others, and there were some good others, but most of them were telling us, wait 2 or 3 years. We can do linear. We don't even know how to do circular. And it's a very different manufacturing process. So, we came to uBriGene. And the other thing, too, and I'll give uBriGene credit, and this is not scripted, just in case people are wondering, at circRNA, we move very quickly, right? So we're doing in a very short period of time, it's taken other companies years. And so we have kind of an internal motto, my motto internally, is, run, nice results, start running again. When we came to uBriGene, we gave them a very they would consider a very short timeline, but they're making it. So, from start to finish to get the clinical, basically taking the initial procedure, making it to clinical commercial scale, and manufacturing first clinical doses, it's going to be about 6 months from start to finish. That's unheard of. Most of the time it's 12, 18. But as Circurna, we want to move quickly. We're going to be in a clinical trial with the yellow fever vaccine by March, no later than March of 27, so less than a year from now. So we needed to move quickly, and they did it. And that's another consideration too, is particularly as a smaller company. And as I said, I had a firm, I worked with hundreds of companies. I've dealt with pretty much almost every CDMO around the world. And I can tell you, as a small company, the hardest part with a CDMO is having them pay attention to you and actually do the work in a timely manner. And I give uBriGene credit for that. Again, not scripted. And that's not always typical. But I think if you're looking to get into, let's say, something like circRNA, it's finding a CDMO, not just that can do it, but that actually is going to do it and do it on a timeline that you find reasonable and be responsive. We have weekly calls with uBriGene. We visited there multiple times and such. So I think, and again, I'm talking more to the startup of small companies. I think those are considerations you need to take into account when you're thinking about who to work with. And, we've been happy with uBriGene. We're on schedule, and we're on circRNA schedule, which is a very fast schedule.
Thank you. Appreciate it. I can certainly say the respect is mutual. Okay, just in the interest of time, one final question, a technical one. Why do you prefer HPLC over chromatography for purification? Yeah, I think it's our question for us. So in strict term, HPLC is chromatography, right? It's just high pressure liquid chromatography. So we use a very specialized chromatography for not only purification but also analytics, as Scott alluded to. The separation is much better between circular versus nicked. So yeah, Scott, do you have anything else to add?
No, I mean, I think you nailed it, Jeff. You know, HPLC is what we use routinely for analytics, and it's an ideal system to use for small sample sizes like you get within process control and lot release testing. But, you know, we're using preparative chromatography for the downstream process development.
Right. Thank you. All right. So we are 5 minutes over time. Thank you, everybody, for staying on. It has been a lot of fun. Thank you, Robert, Peter, and Scott. Really appreciate your contribution. All right.
Thank you.
Bye-bye, everybody. Have a good day.
Thanks, everybody.
Bye, all.
Take care.
Bye-bye.